From Microbe TV, this is TWiEVO, This Week in Evolution, episode 53, recorded on April 2nd, 2020. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Utah, Nels LD.
Hey, Vincent. Good to be with you from LD Home Studios.
That's right.
Yeah, it's been quite a month. We were just sort of reflecting as we were firing up the engines here, how 1 month, it's been a month as the new saying goes, but it feels more like a year or even longer, given all of the current virus-related events we've been grappling with.
I think when we last talked, it was already pretty extensive, but it hadn't really hit the U.S. yet, right?
That's right. And so this was the last week of February, and certainly, I mean, that was what we dedicated the last episode to, was discussing the evolution of the coronavirus, and we'll return to some of those themes and add in some additional new information as that's been coming in. But it's been a sea change, I would argue, in terms of sort of speculating about what this might look like based on limited data or information that we were hearing about, mostly from China at that point. And now it's sort of obviously overrun a lot of the cities in the U.S. in various different ways, and including in your locality. So I wanted to ask you, just kind of at the outset, Vincent, how are things out there in the New York region?
Well, we're all home pretty much. I've gone in maybe once a week for the last couple weeks, and city is, well, first, there's no traffic.
Yeah.
It's like, and all the toll collectors are gone. They've made them go home and everything's automatic. There's no traffic. I get in 50 minutes. The streets are pretty much deserted, although there are trucks on the road and there are people. A lot of people wearing face masks. Our hospital's overloaded, has hundreds and hundreds of COVID-19 patients. And Daniel Griffin, as you may know, has been giving us some updates on what's happening in the area. And, New York is booming in terms of cases right now. And so I'm not sure we've peaked yet, but most people are at home. Schools are closed, restaurants, gyms, all that stuff is closed, only essential places. So it's very weird. Okay. That's what I should say.
Yeah.
Even out here in the suburbs, it's really weird. What about you?
Yeah. So the case numbers are quite a bit lower here. However, this is kind of happening, and we'll talk for a few minutes about some local brushes with some specific contacts who've come back with COVID-19, SARS-CoV-2 infections. But so actually I have a little local dashboard here, a statewide dashboard. This was updated daily. It hasn't been updated yet today, but for Utah, currently just over 1,000 cases confirmed. And this also has some breakdowns both by county. So Salt Lake County, where Salt Lake City is, that's about half of the cases, which makes sense given the population density. And then also indicates how much testing has been happening. So far in Utah, a little over 20,000 tests that have been confirmed. And so a pretty modest number, a pretty small case number. Sort of on a positive note, so far the death totals attributed to COVID-19, the infectious disease, is 7 out of those 1,000. Yeah, but obviously still early days, and so we're also the streets are pretty deserted. Probably maybe a couple weeks, 2-3 weeks, who knows? Still just kind of speculating behind the curve for bigger places like NYC. Obviously, very different. Population densities and just scale of kind of urban population. So I don't think it's going to echo completely, but absolutely listening intently to everything that's happening out there, including the great TWiEVO episodes that you've been posting almost daily. Are you going to have to, you're at this pace, Vincent, I think you're going to have to change it from This Week in Virology to This Day in Virology.
Yeah, in fact, just before this, I recorded another one.
Is that right?
with Jon Udell. Do you know him?
Yeah, Jon is great virologist at the NIH.
Yeah. So we mostly talked about principles of immunology, right?
Yep.
Because a lot of people, I think, would be interested in understanding what this virus is doing, what a vaccine might do, and so forth. It was really good.
Oh, great.
And then tomorrow we'll do another one. So at the end, I say we'll be back next week, and I always feel weird saying that. Usually it's sooner.
Yeah, right.
On Fridays, we have an update with Daniel Griffin. He comes out in the first half hour or so, and he's in the hospital all the time, so he takes a break, and he calls us up and tells us what's going on.
Yeah. I heard, I was listening a little bit last night to the most recent one, I think, where he was coming kind of live from the hospital parking lot.
Yes, that's right. That's why the sound wasn't great, but we're working on that.
No, it was just fine. And if folks haven't already been dialing into TWiEVO, big recommendation. There's some overlap, obviously, with our listeners, but for those who haven't, absolute big recommend to get some of these reports from Daniel, from the physician angle, as he describes the situation as being the hospitals becoming overwhelmed. How he's keeping that positive outlook is a testament to his resilience and
for sure,
and just incredible strength going through some of this stuff. But also, I think it's really great to have these in-depth conversations with folks like Mark Dennison, coronavirologist, in addition, or Jon Udell, as you're saying, immunology, sort of angles and coming at it. The other constellation of this week podcasts, right? So immune, it sounds like you also have, I haven't had a chance to catch up on all of that. I don't know how you record them all. I can't even listen to them all, but really a wealth of information sort of unfolding in real time.
Yeah, I hope people aren't disturbed that we're recording so much. Obviously, you don't have to listen, but have opportunities. And I have guests still in the future. I'm looking at coronavirus people. I want to get in and give their take on it and so forth. I think it's a good opportunity. I mean, I'm home. I am still teaching my course, but so I can record I spend most of the day in my office here, about 8 a.m. to 8 p.m. or so, just recording various things. And I think so as I've said before, our podcasts are popular, but in the scheme of the world, they're pretty much ignored, right?
Absolutely, yeah.
Because there's 7 billion people in the world.
Right.
So, but now is the time when all of them, including TWiEVO and Immune and TWiEVO, get more attention. And I think it's our chance to educate people and give them good information. And the prevailing sentiment I'm getting in our emails from our great listeners is that they can trust us for good, solid science information that they can't find anywhere else. And that has always been my goal. And I'm really happy to see that we can do it, even though sometimes we wander out of our comfort zones, right?
Yeah.
So I think that's why I'm doing this. I've cranked it up because I think we're in a good position to do this and no one else is really.
Yeah, no, I agree. And there's probably too many recordings to catch up or keep up with them all. But even just tuning in, I've noticed for the first 20 minutes, 30 minutes, just at the top of the hour to really get sort of that information from an expert and to have that question and answer can really be a way forward, I think, for all of us as we're trying to interpret the news, the changing scenarios. It feels like it's different by the week, certainly even by the day, almost by the hour. And so, I think that also sort of fits into what we're trying to do here, obviously from a little more of an evolution angle. And we'll talk a bit more on this podcast, this episode, about some of the evolutionary ideas related to SARS-CoV-2. In particular, I think it's important. So some of the news pieces that we're seeing flash out that are trying to launch maybe a half-baked idea that there are all of these new strains or different strains that are mutating. I think we will do a little bit of a deeper dive on that. Before maybe we get into some of the evolutionary stuff, though, I did want to, and kind of referring to some of all of the TWiEVO episodes that are coming out, some local brushes that both you and I have had with colleagues who have had cases, infections with SARS-2. So one of the episodes that's up on TWiEVO now is with Ian Lipkin, an interview you just did a few days ago or maybe a week ago. a TWiEVO special. It's sandwiched between episodes 594 and 595. And I thought it was just absolutely fascinating. So remind me, he thinks he was infected when he was over in China and then came back to New York at some point?
No, so that was a long time ago. He came back from Beijing way at the beginning of the outbreak, and he was quarantined for 2 weeks by the university. And after that 2-week period, he was fine. But then he thought he did a lot of interviews at media places in New York where you go and you have to have makeup put on. You contact a lot of people. I did the same thing. I was on CNN not too long ago.
Yeah, I saw that.
And so he thinks that's where he got infected, basically.
I got you. Yep.
And in fact, they wanted to make sure it wasn't in the lab because he does have the virus in his lab, but they ruled that out completely. And so he got it in the community, as so many other people are getting it. But now he's much better and he's recovering, which is great.
That's great. And so for folks who don't know, Ian Lipkin is actually a virus hunter. So that's why he was over in China, right? I guess was as part of the surveillance of emerging viruses. And so he describes how in his lab, they could quickly move to sort of, homebrewed testing from their own shop. And he was able to confirm his case.
Absolutely.
Yeah. So we've had, obviously, smaller community here in Salt Lake, but we had certainly a shot across our bow. So a colleague of mine, Clement Chow, who's an assistant professor in human genetics, his lab is just a floor below mine. We learned about his case of COVID-19 from a tweet that he actually launched from the University Hospital ICU. And so this was a crazy kind of turn of events a couple weeks ago, which really sort of crystallized it, right? We go from this sort of hypothetical idea of, NBA players, or there's some Utah Jazz players. Rudy Gobert, I think, was the first NBA player who was diagnosed, and this kind of quickly led to the shutdown of the NBA season. But this, at least for me and my colleagues here in the medical school, really kind of brought this to our doorstep with Clement's case. And so I should say up front, he's recovering at home now after a 5-day stint in the ICU, but a little touch and go. So he wasn't on a ventilator. He wasn't intubated, but it was almost, within an inch of doing that, given his oxygen saturation. And then, this was how we found out about his case, was actually from when he was just well enough, still in the ICU, but just well enough to tweet about it. And Clement's a prolific social media enthusiast, so he's on Twitter multiple times a day. But it actually turned out to be really great that he brought this information forward, because so the Department of Public Health was working with him. He was talking to epidemiologists, and they were starting to do sort of traditional contact tracing. But that process, takes some time, both when we consider sort of how the testing system has been overrun in different ways. It seems like every time the testing ramps up, there's another sort of leak in the bucket that slows things down. And between the time it took for the test to get returned as he was on oxygen in the ICU and the information was passed along, that was several days. And probably the most sort of, from Clement's contacts, the most extensive in the timing of it that he could have potentially passed it to others was when he was sitting on an NIH study section just a week before his symptoms started to emerge. Wow. And so that ended up being a lot of conversations with some colleagues who were there and really freaked out about, what was the timing of the exposure, what was their sort of connection to this. And so I won't take too much time to kind of go through all the details, but Amy Maxman, who's a great science writer, actually picked up the story mostly from Clement's tweets, because this was kind of playing out sort of in plain sight in some ways. And she did a news piece in Nature a couple weeks ago. We'll include the link. And this has some quotes and some background information from folks on the study section. And kind of puts into a bigger picture. So not just Clement's case, but also how, as a country, we're still grappling or struggling with things like, testing in a timely way. You still hear stories today about people who might, you can do a test, but returning the results can be a week or 10 days, depending on the sort of pathway that the swabs have to go through. And then, and I think also how, given the way that SARS-CoV-2 transmits, that you can't sit on information over days over a weekend, or if you're part of a contact chain, if you sit on an email, that could really make the difference between someone being able to communicate with their family or change their behaviors to try to limit contacts as well. And so some real challenges highlighted in that piece and sort of echoed around the country in some ways.
Yeah, it's interesting. We're not immune. Scientists can be infected too. We travel a lot. We interact with people. That's one reason why many labs are shut down, to reduce the number of people that are interacting with each other, right?
Yeah, that's right. And so that story is sort of framed, scientists exposed to coronaviruses wonder, why weren't we notified? And I think what's kind of emerging, at least from this corner of our science community, is this idea that we kind of have to make sort of grassroots efforts kind of come together as we get information, share it with each other, take steps, not kind of sit around and wait for directions, but take steps that we think, given the information we have, could be useful and positive. I think it's a balancing act between, not getting too far out on a limb, but still, I think, acting in a timely way, and to kind of trust your instincts a little bit to, really try to get information out there or connect with other folks in order to sort of move through this in a situation where a lot of, I think, the traditional structures and leadership, all the way from sort of the top down to even local communities, could be lots of professionals who know what they're doing, but very quickly overwhelmed given sort of the scope of what we're seeing unfolding in terms of the spread of this virus.
Yeah, it's really remarkable how this virus has basically brought the world to its knees, right?
Yeah, it's really something. I mean, if we just rewind the conversation we had on the last TWiEVO compared to the one that we're sort of, that you're having on a lot of your TWiV podcasts and that we're having today, my goodness, it's just it's a sea change.
Yeah. And as we were saying on TWiV just before, this is not even a particularly lethal virus, right? It's, it's not great, but that's only because so many people are being infected. Could be worse. Could be much worse. Yeah.
Thank goodness for that, kind of a silver lining, in terms of the, what you could imagine the impact would look like, if you look at some of the more pathogenic viruses out there. However, the transmission, I think what we're even learning just in the last week, some of the data as it's coming out, or some of the experimental approaches, and combined with the epidemiology, combined with sort of the patient monitoring. But so some of the notions about this being passed under pre-symptomatic, asymptomatic conditions, the high viral loads that are possible in the upper respiratory tract, that these things are kind of conspiring to really allow this virus to move quickly through and almost silently through populations, which really does challenge, already if we have kind of good testing and contact tracing, even under the best circumstance. I think some of the details we're hearing, it's really hard to contain this thing. And in some ways, I think, if we kind of rewind the tape and look at how China's response so far, kind of some of the really massive interventions they've taken, still leads to a lot of cases, still leads to a lot of death. And yet it does seem like they've really got on top of this, compared to sort of what we're up against currently in a lot of cities across the U.S.
But when you think of the Chinese population and compared to ours, they did a remarkable job, right?
I agree. And I think it's a bit dangerous to, I think we hear a lot in the news these days, sort of this idea of, oh, well, they're just not sharing all of the information. I think that's a little bit bogus. And in fact, if you take a closer look at what they did compared to what we're doing currently, it is sort of almost an order of magnitude, I would say, a more serious level of interventions to, isolate people very quickly, and even from families, things that I think would sort of shock the response, or the sort of our cultural ideas of what a response would look like in the U.S. But it's, and we have to figure out a way forward that makes sense given our system. But there are some real advantages to, I think, these huge interventions in terms of turning the tide much more quickly.
Well, Nels, as we're recording, there's 980,000 cases. We're going to be well over 1 million very soon.
Yeah.
Wow. But, this could infect easily 50 to 70% of the world's population on this first spin around. It's a lot of people. Unprecedented opportunities to study spread of a virus, immune responses, and so forth. Amazing. And I'm looking at the U.S. map. Every day there are more dots.
Yeah, it's really true. It's hit every state.
Amazing.
In some ways it feels like this, massive uncontrolled experiment where we'll see, kind of, it's very complicated, but that, what are sort of some of the themes that you can pull out in terms of how does the virus move through populations depending on the size of the city, the kind of interventions. I think it's one of the exciting things that has kind of caught my eye is how people are thinking about, kind of tracing, not in sort of the traditional classic epidemiologic way, but based on things like our cell phones and hopefully anonymized data. How much are people moving around? How is that behavior changing? And then as this continues, how do you sort of correlate those differences in movement, both in time and space, different locations, and then see what that means for how the virus moves through populations?
For sure.
Yeah. So I wanted to I think we'll dive into a few evolution-focused issues, but I just wanted to highlight a couple of links that have been really, I think, useful to me and sort of in the spirit of science communication, I think, stepping up to the plate a little bit. And so, to underline, I think, what's happening on TWiEVO, these great long-form Q&A conversations where you all are entertaining the questions from I think it sounds like you have hundreds of pages of questions in the kind of a backlog.
It's amazing.
Yeah.
I get over 100 emails a day.
Yeah, that's really incredible. I think it highlights, right, that people are really are hungry for information that we might traditionally think, oh yeah, we'll have a organized response from the CDC or other sort of professional leadership of governmental institutions. And there's a little bit of a vacuum. And so, and this is kind of what I mean by some of these kind of grassroots approaches where we can step up and we're not experts in everything, but we'll, sort of take this scientific view, have questions, adapt as new information as it comes out. So I think, one of the things that has been really inspiring to me is listening to Rich Condit saying, when we were talking about this a month ago, we had no idea what we were up against. And instead of sort of digging in and saying, well, it's, this is what I thought and this is what it is, to really say, no, I think in Rich's word, we're going to school on this and we're updating as we go. We're acknowledging that we're going to get some stuff wrong, that we don't have a full reach. But that, I think, for me, highlights the importance of kind of a scientific or medical approach to this, that the conversations can happen in real time, that we can adapt and adjust to new information.
What I find interesting, a number of universities are starting to have Zoom conferences for the public, right?
Yeah.
Where people are presenting things and they want people to listen. So I said the other day, oh, this is what we've been doing for 12 years. They're finally getting it, that people want to hear what you have to say.
Yeah, that's right.
Why does it take a pandemic for you to do this? Why don't you just do it, once a month, open up a Zoom to the public? They'd love it.
Yeah,
right?
That's right. And that's, something I'm hopeful for is on the other side of all of this, whenever that is, that that hopefully those, the kind of stress test here will show those, places where some simple investments in things like this could be really beneficial for strengthening that bond between the scientific community and the general public. So anyway, my endorsement to keep going and maybe to, just go a little farther on the scientific approach. There's a big difference here. So if you or Rich or Kathy say something that's just patently wrong, your crew isn't going to just say, oh, that's great, you're doing such a great job. You'll correct the record. And,
yeah, of course.
Yeah. And so, but that's a little bit different than what we're seeing, I would say, in some governmental situations here, where you can't, question the authority from above. And so science is arranged so that the authority doesn't come from any specific person. It comes from the evidence. It comes from the information. And I think that's so important. now, especially as this thing is really moving and causing some really dire global and public health consequences, as Daniel Griffith was describing from the hospital parking lot, as they're overwhelmed currently in New York and in other places. And this is kind of coming down the pipeline in other towns and cities across the country as well.
What I think is unusual about our forum, Nels, is that not only do our colleagues correct each other, but the listeners call us out.
Yeah.
They say this is not right, and we accept being corrected. It's fine. You can't find that anywhere else. You can't do that on CNN or Fox or whatever you listen to. In fact, it's a one-way stream from them to you, right?
That's right.
I think two-way is the way to learn.
Yep. So then a couple of other links I wanted to lift up. One is, so, as I get more questions as a virologist about what is the coronavirus, how does it work, how does it infect cells, how does it move through cells, how does it replicate? One resource that I think is great is from a really good colleague, Britt Glounsinger. She's got her lab at UC Berkeley, and she posted on YouTube maybe a week or two ago a Coronavirus 101 lecture. And this, really starts to go into some detail about how coronaviruses work. And we talked about this a little bit on the last TWiEVO. Coronaviruses are sort of if you kind of in the whole span of virology, have been somewhat underappreciated compared to the level of attention that they're getting now. The number of labs devoted to coronaviruses is pretty modest. There's some great folks that you can hear on TWiEV. So, Mark Dennison, an example, Ralph Barrick, so the multiple interviews that you've done with him now, really enlightening. Same with, I would say, Matt Freeman at Maryland, Vineet Manachari at UT Medical Branch, a growing community, but still on the whole scheme of things, a pretty modest group. And so having some of these connections or sharing some of that information and understanding how these viruses tick, I think is sort of an important part of the conversation as well. And then I had a few kind of favorite science communicators I wanted to mention. So Ed Young, he runs, I think, the science page at The Atlantic. Big recommendation to check out the writings that's happening there. Liz Neely, I think, have you come across her before, Vincent?
Yeah.
Runs this, is it the Story Collider, I think?
That's right.
Yeah. She had a really nice short article, I think in The Atlantic, about science communication, how to reach audiences, and how to reach the general public, something that I was definitely tuning into. And then, as I mentioned, Amy Maxman, who sort of lifted up this story from our community, and she's out there really trying to sound the alarm in terms of a lot of the ongoing shortcomings for viral testing and for contact tracing.
Good stuff.
Yeah.
That's, of course, just some of it. Ton more.
That's right. The tip of the iceberg. It's a good point. So maybe we should spend a little time transitioning into some of the evolutionary issues related to SARS CoV-2. We actually have a paper. We'll see how it times out. And I know, and just let me know too, Vincent. I know you're have a full menu of podcasting, and so if you need to step away at any time, no problem. But I think we'll run through some more of the kind of evolution-focused points on SARS-CoV-2. And then we do have a paper on a whole different a virus that was just sort of being glimpsed for the first time. We had this in the queue even for last TWiEVO. We didn't quite get to it, and I wouldn't be shocked if we're sort of delayed again. But there are other viruses out there. There's good reason, certainly from an evolutionary standpoint, to consider a lot of this diversity beyond the current sort of virus events that we're grappling with. And then other evolutionary stuff. And we'll kind of start to, I think, hint back at that as we get into the some of the questions from listeners later in this episode. But so one of the points that I think both you and I are seeing is this idea that as, some of the frontline genomic analysis of SARS-CoV-2 is starting to map SNPs, or single nucleotide polymorphism. So these are the point mutations, the A, T, C, and G. Or I guess we're talking RNA here, aren't we? So the substitute the T for a U to the uracil and the RNA base. But that these, as mutations accrue, which is what happens with viruses. Viruses mutate. The coronaviruses, as we discussed, are relatively modest in their mutation rates. That probably reflects that they're for an RNA virus, a really massive genome, sort of the largest class of RNA viruses, actually. And so actually accruing a relatively modest number of mutations, but still quite a bit. And there's been, I think, in the news now, some troubling misinterpretations of how to interpret what this variation means, sort of in a couple ways. So one thing we hear about is that there are now all of these different strains circulating that could have different pathogenicity, some that could be worse or that might be sort of more dangerous than others. And that's not a new thing. So I heard, I think this even around the time when we recorded a month ago, there was this idea that they're the so-called S in the L strain. Did you see that one from a while back? Yeah. And some people are saying, oh, the S must stand for safe and the L must stand for lethal. Total red herring. So, so that definition, actually, it was based on a single nucleotide change, one mutation. And we'll talk a little bit about this next strain initiative out of Seattle. It's run by Trevor Bedford. But so he took a quick peek at that a few weeks back and noted that that designation was based on a single mutation. And you could follow actually the, just based on even earlier sampling of virus genomes, what percentage of viruses had the one mutation versus the other. And it was about, I think it was like 40, 60%. And over several weeks, it stayed 40 or 60%. And so at least, so what that suggests, so first of all, the idea that a single mutation in 40,000 sort of possible positions, and then you have to remember all of the myriad combinations to get to something that could have a functional consequence versus the majority of mutations, which will either be, at least according to evolutionary theory, slightly deleterious, meaning the virus might replicate even more poorly, or neutral, meaning that the virus still works just as well, but not better or worse. It's just you can tolerate, there's relaxed constraint, or you can tolerate different changes and still undergo a replication cycle. The thing that evolutionary biologists would be looking for, if there really were sort of functionally consequential mutations that would allow a virus strain to outcompete another, would be that percentage, the 40-60, might change very quickly to 50-50 or 70-30, and then 80-20, 90-10, and then 100% zero. And that would be indicative where basically a single mutation would sweep through a population. All of the other viruses that didn't have that mutation would be lost because they'd be outcompeted. That would be sort of the definition of a hard sweep. We haven't seen, to my knowledge, there's no evidence of any kind of consequential sweeps going on outside of sort of the usual amount of noise between sort of the frequency of mutation sort of in any combination.
So during SARS-1, classic SARS,
yeah.
about halfway through the outbreak, a variant emerged that had a deletion in one of the genes encoding a protein called ORF8, and that swept. That was then in all subsequent isolates. And so immediately it was suggested that it was increasing the virulence of the virus, or maybe the transmission. I don't remember what. It turns out it actually reduces replication in cell culture, and it's been postulated by Christian Drosten and others that maybe that was selected because it lets the host live longer and transmit better, because really transmission is the major selection put on a virus, right? To find a new host. So that's an example where even if you do see a sweep, it doesn't mean that it's detrimental, right?
Absolutely. So it's beneficial in that case to the virus that has that variation, in this case, the presence absence, right, of ORF8. But yeah, it's exactly the right point that it's the competition here, and that can, in some cases, depending on how that virus is moving through the population, that could be linked to pathogenesis, if that somehow allowed transmission to work better. But those aren't those don't correlate 100% or don't need to be linked together. And in many cases, I think in evolutionary virology, the idea would be, if anything, you end up attenuating, because as you're saying, if you kill your host quickly, it's very hard to replicate as a virus and then to spread. This is actually a way that viruses can burn out really quickly, as opposed to transmit over longer courses of time.
And part of the discussion has been, well, will this become more transmissible? And to which I say, something is selected, something appears if there's selection for it. Right now, this virus is transmitting really well. So right now, isn't that a good one way to look at it? There's no selective pressure to get better at that property.
Yeah, that's right. I mean, we're still, that's a point of active investigation. And what's really, I think, tricky about this one is that, so first of all, as we're still trying to catch up on testing at a scale that approximates the massive number of individuals in our species, and then also, some of the features that are coming out that we've already been discussing, the fact that this virus can transmit so well, it's already, so in one sense, one way of thinking about it is sort of the really consequential of evolutionary events already happened. When this virus was sort of smoldering, whether it was in a bat or there was some other intermediate host that still is somewhat mysterious, that's sort of when the consequential evolution happened. Somehow, as mutations were sampled and sort of spillover events were potentially happening, they're in that 40,000, or remind me, how big is the genome? Is it 29,000?
30,000, yeah.
30,000, yep. Base pairs, some combination came together that is indicative of how well this thing can spread today. And so in that sense, the evolutionary action might have already happened sort of, in the last months or last several years, maybe the last decade, as this thing smoldered and then emerged. And so in that sense, I would say that this virus, SARS-CoV-2, has already hit the jackpot in terms of being really good at spreading through the human population, as we're seeing now in real time.
And it's worth comparing it to MERS coronavirus and SARS-1. SARS-1 we were able to get rid of because it didn't transmit very well, and mostly it transmitted in very sick people, so they could be contained in hospitals. So that was easy. And so even though that was able to go person to person for extended chains, eventually we could stop it by interrupting. And then there's MERS coronavirus, which is never able to maintain very long chains of infection. It always fizzles out. So it's never achieved that perfect combination that you were mentioning of, and maybe it never will, because it's been now since 2012 or so. Yeah. And, short chains of infection from a camel to a few people, and then that's it. So apparently it's not going to achieve that, right?
Yeah, no, I agree. And so if we're thinking about this kind of from an evolutionary standpoint, so obviously there's massively important reasons from a medical standpoint for us to have our attention focused on this strain. But from an evolutionary standpoint, I would make the case that we should really be thinking about, well, what's the next one that might be lurking in the bat populations? What's going on in terms of how many strains are close to doing something like this one is doing now? And so how do we sort of refocus at least some of our efforts into, preparing for what we might see, 5 years from now, 10 years from now, who knows exactly what the tempo might be. And part of the reason we don't know is because there's actually a pretty modest amount of surveillance, relative to sort of this last minute, okay, we're dealing with a health emergency here, as we have already been discussing.
I mean, I would argue that we should look in a lot of other animals besides bats, right? Because they're all candidates for harboring viruses that can spill over into us. We barely sample bats, right?
That's right.
It's not easy, but not. And the pangolin was, I think, the only other animal that had ever been sampled in which SARS-like viruses were found. So I would say, an ounce of prevention is worth a pound of cure. Now we're paying trillions of dollars to catch up. And can you imagine what that money would have meant in terms of fundamental research in all aspects of this, right?
Yeah, no, of course, I 100% agree. I mean, hindsight is 20/20, but yeah, that's right. the small amount that we invest regularly for basic science or for trying to understand these patterns, or the molecular epidemiology, the ecology of the viruses, the investment is so little compared to what we're up against now in terms of having to respond once the kind of horse is out of the barn, so to speak. Yeah, it's really striking, and hopefully, again, as this thing goes through in the coming months, that on the backside of this, will this be another opportunity to have that discussion again and try to bring some real resources into taking a bit of a longer view than sort of this responsive view that we're stuck in the middle of right now?
I hope so. I don't have a lot of faith that it'll be much different.
Yeah.
We have the technology to have prevented this, essentially, if we had had the right antivirals and maybe even broadly acting vaccines. We could have done that and been ready for this, and the reason we don't is there wasn't enough investment. And, there's a philosophy of government on the one hand, which is to put the minimum possible into everything. And, considering that science funding is not great, the fact that we are able to respond amazingly, the science and clinical communities have been amazing in what's going on, is incredible, because we know how to do things. But with more money, we would have prevented this entirely, I'm quite sure. So I hope you're right, Nels.
Well,
I hope that we'd say, all right, let's fund programs, pandemic preparedness. Let's have a plan for face masks and ventilators and so forth. On the other hand, it really depends on the politics that run everything, and I'm not sure that science will overcome that.
Yeah, no, I hear you. And there's certainly historical precedent that humans will fall back into patterns. I mean, if we think about some of the past massive influenza pandemics and then sort of the limited ongoing responses, if we think about that we're heading towards at least a recession, maybe more, the economic resources are going to be spread thin, because a lot of the research we're talking about is linked to medical schools and all of the pressure that's being put on hospital systems right now in terms of revenue streams. It's going to be really hard to continue to make the case, but I think we need to, and hopefully there are a few cracks or the ability to get through and to try to bend the curve to some investments. But yeah, that's another sort of major piece of this that I think is just getting underway. But so anyway,
can
go
you
ahead.
Explain this nextstrain.org, what's going on there?
Yeah, thanks for bringing that up. So, and this actually this builds out of, I think, exactly this idea of, people proposing that there are all of these strains now circulating. So NextStrain, this is and we'll put the links up. It's actually worth going through this. So if you're listening, maybe pause the podcast for a second and actually open this link. We'll kind of step through it sort of in real time here. So this is outfit based out of Seattle. One of the major sort of players here is a fellow Trevor Bedford, who's on the faculty at the Fred Hutch Cancer Research Center. And because, it was sort of this early collision, I think it was even back in, mid-January, that one of the first cases was identified near Seattle, and it's associated with this in Kirkland, with this retirement home. Trevor and some of his colleagues were really well positioned to sort of pivot on their usual what they call genomic epidemiology. So this is almost real-time sequencing of virus genomes. They were doing this in regard to mostly focused on seasonal influenza. So that's been obviously kind of lost in the mix here, but we're also on the tail end of a pretty impactful influenza season. And so they pivoted, and there's a group, the GISAID, I guess, or GISAID, I don't know how you pronounce that acronym, out of Germany, that also collects virus genomes kind of in real time for this genomic epidemiology. And so this is now at NextStrain at this link for the coronavirus has been kind of retooled to really try to get a really high resolution view. So what you'll see on this link is basically what looks like an evolutionary tree. And I think this is where some of the confusion probably really arises, is that when you look at trees of life, so this might be, a set of mammals might be shown in one clade, which is, another word for a collection of species that are closely related to each other. And then you might go over and you'll see the reptiles are in another clade. And so those are when we look at these trees of life or trees of different species, it's very intuitive to think, okay, in this one grouping, that's one kind of animals, and this other grouping is another kind of animals. It has a little bit of that flavor when you look at the virus strains. But there's a big scale problem here, or difference in scale that you really need to pay attention to, because here a clade can mean a difference in a few of these point mutations. Mm-hmm. And whereas the difference between a species in the millions of years of divergence can be a million of these point mutations. And so when you just look at the virus tree without any scale, and you see those collections, those different clades, you might think, oh my goodness, there's a dozen different strains circulating. And in fact, that's now kind of infiltrated into the news stream. However, if we go back and look at the scale, and you can do that actually at this website. So it's worth playing around a little bit. So you can first of all change just by a click of the mouse how you arrange the tree. So there's that rectangular sort of left to right view. You can go to the radial or the unrooted. You can even do this on a clock, so you can get a sense of how many over the weeks here, how many point mutations you would expect given the sampling of the virus. So if you click on the clock, they currently are estimating a rate of 24.5 substitutions per year. For a virus, that's actually not really remarkable at all for an RNA virus. So HIV, influenza would outstrip that rate estimate. But because we're looking at it on a tree, I think it's a very natural, intuitive leap, basically, to think, oh my goodness, these are different strains. Mm-hmm. If you actually put your mouse over the dots, which is each dot represents a different virus that was recovered from a patient, and then its genome sequenced, you can actually kind of learn about them in real time. And that's one of the really, I think, the advances here is this resolution that you can get. And so are you at the website right now, Vincent?
Yeah, sure.
Yeah. So if you look at that top, so if we're all looking now at the kind of the big tree here, right in the middle, do you see this sort of big concentration of red dots?
Mm-hmm. Sure.
Yeah. So if you go to the very left one of those and put your mouse, hover that over, this one's called USA Washington 1, 2020. And it's got the date on there. So I think it's the 19th of January. Yeah. And that was the first strain that actually showed up in that nursing home in Kirkland, Washington. And then the dots to the right of that, it looks like a clade. It looks like its own strain. That's sort of a false sense of what's going on here. These are there's a concentration. So because they knew that was being spread in the nursing home, they now have done a lot of testing in that really little community. I mean, it's literally just one single building. And then if we go out to the very tips of the branch, so you can go if you do the sort of the top one on the tip, this virus is called USA Washington 375. So now in that chain, this is the 375th patient through that sort of sequencing platform. I wouldn't be surprised if this is someone in that same nursing home, and we can then you can actually compare the mutations. So and count them based on the scale. And so what we're looking at here is actually between 5 and 10 mutations away from that one that was back in January, and on that sort of that time scale. And so a couple things, I guess, take-home messages is to realize that this is like the scale here is all different from how we usually think. Yeah, about trees.
This is not a cat and a dog, man.
That's right, exactly. And in fact, if you put all of these virus sequences on a tree with a cat and a dog, so because if you have a shared, you could use the polymerase gene, perhaps. Yeah. All of that spread of this tree would be one little dot compared to the length between the cat and the dog, and yet we just don't quite have that context yet. And so that's what that would be my take home for today's TWiEVO is the scale on trees. And so for that cat and dog tree, there'll be if you look in the corner of a lot of phylogenetic trees, there's actually a scale bar. And generally that's not explained in enough detail. So it might be like a centimeter long line, and it might have a number like 0.1. And what that generally means is that there's 0.1 substitutions per site as represented in that length of a tree branch. And so that gets a little bit complicated because that number in the algorithms that actually draw the trees, it's averaging the differences over the entire gene or whatever is the characters, the set of characters that are being compared between the species on that tree. And so for a dog and cat, you might have a centimeter as 0.01 substitutions per site on a sort of a reasonable tree. For these viruses, that scale, which isn't shown in the same exact way, might be 0.00000001 to sort of get to that same level of differences that you see that's just accentuated so that you can see these minor differences when you're just comparing it between the viruses.
And the other cool thing here is that they've colored them by country. And so, purple was originally China. And then when it got to the U.S., it's colored red, right?
Yeah, agree. Yeah.
And then you can see where the red is going. Then you can see the red goes to other countries.
Exactly.
So it's spreading from the U.S., right?
Yeah, exactly. This is a really good point. So this is how, so if you scroll down from the tree to then the world map, this is how they make these predictions. And the other thing to remember here is this is a hypothesis basically, right? So there's evidence supporting, if we look at these world maps, you have that color code and then these lines between locations, right? So the purple sort of circle in China that then moves over to the US where it becomes a red circle. So how is it that they actually make those inferences or generate that hypothesis, claim that idea? And it's actually, so that's converted from the tree. And so, for example, if we're looking at that sort of big collection of red viruses that come out of that, many of them coming out of the Seattle area, and then trace that back to the left, now all of a sudden it turns purple. And that reflects the fact that this thing emerged in China. And then based on how these things are related to each other in the tree, how many shared mutations they have in sort of similar patterns, that's what gives the evidence for why you would say, okay, well, let's draw a line specifically from this strain in China to this one in the US, because basically now it arrives in Seattle and it looks almost exactly the same.
So this is basically the most likely scenario, right? Because that's how these calculations are done on maximum likelihood.
Exactly right. So it turns out the viruses can break the rules. They don't necessarily want to tell us all of their secrets. And so in sort of the simplest form. And so there could be kind of cryptic collections of mutations or back mutations or related things like that, which would sort of cloud the picture. But that's exactly right. So this is based on the information that's gathered so far. This is sort of the best interpretation of how these things are moving. And then as you kind of, if you take that sort of deeper dive, then you can start to do more fine-grained predictions about, okay, this virus is in Seattle, but now it shows up in Utah, or it shows up in Minnesota, or it shows up in Wisconsin, based on that chain of custody, in a sense, of the shared mutation and profile or relatedness. The more related they are to one that's been placed in a location, the stronger the prediction that you can sort of track where it's going. And so this, it's, sorry, go ahead.
The other thing I wanted you to clarify is, so this first red isolate from Washington, and then all the reds, most of the reds are then in this big cluster. So can you explain, how we have a founder virus coming in and becoming a cluster on its own? Why does that happen?
Yep. No, and it's all kind of relative, right? So you could start as a reference would be that first Washington strain. And then if you sort of build from there, then if you set that as a reference, then you can start to say, okay, where is the next one that's, so you take patients around the country or even around the world and say, what's the next closest related virus? And it will fall into that category. So if you just scroll over, what you'll see is that a lot of those red dots in the middle actually are from Washington. But here I'm just scrolling over one. Here's one from Canada. Scrolling over, here's one from Minnesota. And what we're seeing there is the probably an echo, basically, of people traveling and then transmitting the virus. But it kind of goes back to that single point at the beginning. for the arrival in the U.S. And I think there's enough known about that kind of from the classic epidemiology that the idea was, I believe, that someone was flew in from China and then started this sort of chain from a single event of transmission through the community. But there's another point that you're bringing up here, Vincent, that I think is really important, which is a tree is only as good as your sampling. So the fact that we see this big cluster of red in the middle is in part because there was such good, sort of a rapid response in the Seattle area to very quickly sequence the whole genomes of these viruses from patients that were coming in and sort of associated with sort of the next strain and then some of the related public health efforts that are happening in Seattle. And so, in a sense, you might look at this tree and say, oh my goodness, there is just like this massive collection of stuff that's happening in Seattle. But relative to the world, like most of the stuff that's happening, we don't even see. It's invisible to us because there's so many people. First of all, we don't know that they're infected, or if they have symptoms, they're just not getting tested, or even if they're getting tested, we're not doing the genome sequencing. And so that's another thing I think, or a note of caution in interpreting these trees, is that there's going to be, in the early days in particular, the sampling is really biased or sort of focused in places that have the ability to do this kind of high level of analysis. It's going to sort of necessarily skew our view of things based on the fact that it's a really small sample that's not sort of balanced across what's happening even in a single city, certainly not across a country, and not even close to across the world.
You can also choose to display this not by time, but by country.
Yes. So the color coding, you can do it by country. You can also do it by time. And then there's another little tab here to do it by divergence.
Mm-hmm.
This is
important. That's what I meant. I've selected divergence and that makes a very simplified version where you don't have as much crossing over of the colors.
Yeah, exactly. And I think what that also illustrates, I really recommend doing that, is it how few mutations are represented.
Yes. Most of them are like between 10 and 15 different, right?
Exactly. And so, this is these viruses that have these like different positions on the tree. This could represent 10 or 15 mutations, whereas a cat and a dog on a tree of life or on a species tree, this would represent millions of mutations. Again, sort of really, I think, nicely illustrating the difference in scale in terms of the drawing of these phylogenetic
But I like the divergence because that takes away the time factor, and it's just like, how related are these sequences to each other, right?
That's right. Yeah, exactly. Yep.
But on top of that, they have the color, so where these isolates were made, right? Mm-hmm. And so you can clearly see when, there's a red one from U.S. or related countries showing up somewhere else, it obviously came from travel. Or, as you say, it may have tricked us. That can happen, I suppose.
Yeah, that's right. And so one of the things that has been really great from this crew at Nextstrain is actually they're doing a weekly kind of a digest or trying to do like a summary of what are they seeing in the past week. It's a lot of information. So I've shared this with some of my friends and family who are not scientists and to get a little bit of feedback. And so it's sort of like page captures and then trying to illustrate some of these points that we're discussing in more detail. It can be overwhelming. It's a lot of information, but these digests and some tutorials about how to read a phylogeny, how to read a phylogenetic tree, a useful start to try to kind of understand this at deeper levels. And then the digests are also including some really practical information about kind of, okay, here are the patterns. What are some things that we can do individually to try to slow down the spread of this virus? And so I'll try to add that link as well to the weekly sort of capturing what they're seeing through the Nextstrain sort of clearinghouse here of genetic information.
Mm-hmm. So you have a comment here from someone who wonders if they're going to have antigenic variation, right? Is that right? Or is it just seasonal reemergence?
That's right. Yeah. So I've been, one of my colleagues, Rob Unclis, who's at the University of Kansas, he's an evolutionary virologist. He actually works in Drosophila, at discovering new viruses. And then also the immunity, using the fruit fly as a model system. How do the immune responses sort of evolve and co-evolve or evolve in these conflicts with viruses? And so he was reaching out, and I was asking him, what's one of the questions he's hearing as a evolutionary virologist? And so he has a lot of colleagues, families, and friends, asking that. So will SARS-2 as we're, moving through the spring and into the summer and beyond, is it with us now for the long haul? And will it reemerge seasonally, like the flu, or migrate like the flu does? Is this with us now for the long haul? So you've been talking some about this or not even just a little bit, but a lot on TWiEVO. I'm curious to get your impressions or how are you answering that question as it's coming up on?
So, in fact, what they should have asked, will it become seasonal like the seasonal coronaviruses?
Yeah, right.
Because we have viruses in the same family that do the same things but people don't think about them because they're so mild that they don't get any press. However, there are 4, endemic coronaviruses that cause very mild infections. And here's the cool thing, and this is all from Ralph Barrick and a little bit from Jon Udell. They infect kids, and very little disease is in kids, and we don't know why. So kids, when they're young, these viruses infect them. They get some immunity. It doesn't last. But then when they get infected older and older, it's a way milder disease because they have some immunity, even if it's not lifelong. And the problem here is, of course, we're all getting infected at once. This is infecting older people who shouldn't be getting that first infection. It should be a younger kid, right? So Ralph and others have said, yes, this is going to become the 5th endemic coronavirus eventually. And this is probably how those other 4 emerged. Years ago, they caused a pandemic and at a time when we didn't even notice it. And eventually, after infecting for a couple of years, maybe enough to infect the entire population, they became these endemic viruses. So that's what we're telling people. They will definitely emerge, but it's going to be a lot easier. We're not going to have to shut down the world every winter.
Yeah, that's right. So, and remind me, so this was like in the 50s or 60s, were those like the first wave of what became these viruses are now sort of attributed to common cold symptoms?
Well, we discovered them in the 60s.
Okay.
We discovered 2, and then came SARS-1, and then after that they started looking and they found 2 more common cold SARS. But the estimates are that they emerged from bats hundreds of years ago. Yeah. At which time, the world population was much smaller. There was no medicine of any sort. So if there were an epidemic, no one would have noticed it. Yep. And they eventually settled into this pattern, which probably doesn't take more than a couple of years. Mm-hmm. Mm-hmm. And so, yeah, this is a new endemic coronavirus. I don't know, maybe next year, or whatever, but I think that's where it's going. I mean, this is based on what the coronavirologists are saying, and I think that makes a lot of sense.
Yeah, I agree with you. Yeah. I wanted to ask as well, Vincent. So we're hearing even kind of in the last few days some new ideas like trying to linking the virus dose, so the amount of virus particles that you might be exposed to in any one sort of given contact, to the severity of symptoms. Is that kind of hit your radar screen, or what's
totally.
Your thinking?
I don't know why. I don't know. I think this is coming in part from healthcare workers who are going to get assaulted on a daily basis and maybe with more virus than other people would. And there was some anecdotal reports that they were getting more serious infections. But yesterday on TWiEVO, Alan gave us a reference where they're not getting more serious infections than anybody else. Now, I think they could get more. A higher fraction of healthcare workers could be infected, but there's no evidence from what I can see that it's more severe. The idea that the dose could lead to a more severe infection makes sense hypothetically, because you can imagine overwhelming the innate response so that then you get a very rapid and progressive disease. But and we know dose matters, right? Mm-hmm. Mm-hmm. But whether it's severe or not, I'm not sure we have any evidence for that. So I don't know why people are interested in, but we get a lot of emails on that for sure.
Yeah, that's what I was wondering. I mean, for me, thinking about just the virology, right, is the dose, sort of the order of magnitude of the dose might not be too different, even in sort of a severe versus a less severe kind of encounter or contact point. But the virus itself, once it's on board and it starts replicating like crazy in cells, now we're talking orders of magnitude like beyond in terms of sort of ramping up.
Yeah.
And so that seems to me like maybe a counter argument to the dose sort of correlating perfectly with severity. How about, I wanted to ask Vincent about sort of more of the practical measures, just as you're kind of correlating, or kind of bringing together information from many sources. So what are you thinking on some of the practical measures that are getting more and more sort of prioritized? Things like, should folks in the U.S. be wearing face masks? How should we be thinking about this?
I started wearing face masks when I go into the lab, just when I'm between my car and, walking on the street, getting in the elevator. And there's always someone that comes in the elevator with me, and that's less than 6 feet. So
yeah, I hear you. Yeah.
I found an old face mask I used to wear when I was changing the insulation, the fiberglass insulation, right? And I wore that. And then at the lab, they had given us each a face mask, a surgical face mask. So I find that a little more comfortable. So I would recommend, if you can get face masks, to wear them if you go out. I went to the post office. I wore a face mask. I think you need to be cognizant of anything you touch being potentially contaminated.
Yeah.
So face masks, stay away from people and wash your hands a lot. And when you buy food or anything at a store, I would try and wipe it when you get it in the home. And I leave my shoes outside. I wash my hands a lot. And I think if you do all that, you're probably going to be okay.
Yeah, it's interesting. I mean, that's certainly, in comparison, some of the measures in China, in addition to sort of like real-time temperature taking, right, as people were going to the grocery store, and then, almost instantaneous quarantining, the sort of vast adoption of face masks, even before the coronavirus, right? But that's something continues to be in wide practice there, but I think is in really limited practice in the U.S. So certainly as I step out, right now I'm on kind of a once a week trip to the grocery store calendar. And the last time I went, I'll confess, I didn't wear a face mask, but I was wearing gloves just because I've been washing my hands so much, et cetera. And so being in a lab, we have a lot of surplus gloves. We're donating most of them currently as PPE to the hospital, as inventories are, people are worrying about that. But even, wearing nitrile gloves, I was getting some funny looks at the grocery store. And I just think as a country, we got to get over that. We got to,
Totally.
Yeah, let's wear gloves. Let's wear masks. And it doesn't have to be an N95, but, something around the house, or all of the DIYs that we're seeing. I would put in a strong case, and let's try to circulate this is let's just wear some PPE. Make sure that we're doing our best to, not take away from the stocks. It really should be prioritized to the healthcare workers, healthcare providers. But as we can cobble this together, every little bit helps, I think, in terms of some of these practical measures that you're describing. Well,
I heard George Gao on, he was being quoted on TV the other day. He's the head of China CDC. You may know he's an editor at PLOS Pathogens, right? So he's often sent me papers to review, but he said the reason for the difference in the U.S. and China is that in China, most people wear face masks, and in the U.S. you don't. And, back in February, Rich Condit and I were kind of making fun of it, but I'm converted. I think it can make a difference. I mean, you look at the data, it can make a difference. And if you can get a hold of some, get it and wear it for sure.
Yeah, no, I think that makes sense. So another point I wanted to bring up was sort of the concept of exponential growth and how kind of counterintuitive this is. So, I've got some friends who are doctors, my brother, who's a surgery resident in California, and just sort of, anecdotally hearing some of these stories. So friends in Minnesota, Utah, places that are a little bit off the current front lines of the peak of these viruses and of the coronavirus. And in some cases, there's, because it's playing out over weeks, over months, there's sort of time, a little bit more time to prepare in the middle of the country or in other places.
Yeah.
And what you see at a lot of doctors' offices or hospitals is actually very little right now. It's almost like a ghost town. It's eerily quiet. And you can even have some friends who describe it in like long bouts of boringness as, for example, elective surgeries are sort of tabled ahead of time to sort of clear the decks. And yet there's, I think, this really almost counterintuitive idea of exponential growth where that's exactly what you'd predict, right? Is things will be really quiet, really quiet, really quiet. And then, you might find yourself like your TWiEVO contact your colleague Daniel Griffith in the parking lot of the hospital with some really harrowing stories about a healthcare system that's overwhelmed in New York City. And so, I'm always curious about how to maybe communicate that, or if you're seeing anything that sort of helps to really illustrate some of those points.
I haven't seen anything, but I think you can use like R-naught to do that, right? If you say every person who's infected can infect 2 other people, and then they can infect 2 other people, and then all those people, and you could make a picture. You could start with 1 bacteria and divide and divide. It's the same idea, right? Yeah. Eventually, if you have enough of these all over the place and they're all infecting 2 or 3 people, then you get this exponential spread. And if it takes about 2 weeks, right, to incubate an infection, you can imagine there's a little bit of a lag initially before you see anything. And then, 80% of the infections are mild. They never show up on anybody's radar.
Yeah, exactly.
So till we get above that, till we get into 20% where people are starting to go to the hospital, you don't even notice it. And by then, it's too late. Mm-hmm. You've got hundreds and hundreds of people. And, we could have picked this up if we had been testing back in January, but we didn't. But that's how I view it, right? Mm. You got one person doing 2, and then each of those are doing 2, and then each of those 4 are doing, so then it really increases exponentially. Maybe a picture. That's the way, that's different from how viruses usually multiply, right? But that's how the infections are transmitted.
Well, and I think actually that point that you're making too about the 80% being mild or sort of under the radar, that adds another kind of layer here where I think that,
you don't see it, right?
Exactly. And then that's almost sort of like a second shoe dropping in terms of the potential of being overwhelmed. It's sort of like it's right. happening all around you, and then it just sort of bursts.
Yes. Yeah. So right now in your state, there's probably lots of infected people, and we don't know much. We're not testing. They don't have symptoms. But as soon as there are enough, and I don't know what that number is, then you're going to start seeing the really sick people, which is the 20% of them, right?
Yep. That's right. And I think that it gets to another facet maybe of the testing that we haven't really talked about yet today, but is the serological testing that's coming online. And so, currently, given some of the bottlenecks or some of the trade-offs of running a lot of the testing through private companies that might have, I'd say revenue-based incentives that might not be the best for public health, we have there's a sort of a massive prioritization of virus testing for folks that do have the symptoms in that 20% category. Now, as serology comes forward, and so here, you're not testing for the presence of the virus, you're testing for the presence of an immune response that has already occurred by the hosts, the antibody response. That gives you a pretty good clue that someone may not even be infected. In fact, the infections pass relatively quickly, but the immune responses persist. And so the serological testing gives you that view on that 80% or 100% in a sense, right, of everyone who has been exposed to that virus based on the specificity of the antibodies to this new virus. And so you have a pretty high resolution view of that. And so I think that really starts to illustrate how we get on the other side of this in terms of the behaviors. So how do you come out of the sheltering in place, the stay home, stay safe kind of campaigns that are happening across the country? It's really, I think we're depending on the serological testing, and now that sort of coming online in a high throughput way that could help to sort of get us out on the other side of this in the coming months.
Yeah, I think that's critical to know who's infected or not, especially for healthcare workers, right? So they can feel a little better about going to work and not worry about being infected. But also we get a sense of how many people are actually infected in the country, and that can inform decisions to let people go back to their normal lives, right? Exactly. Because despite what you have heard from the leaders of this country, we just can't say go back to work in 2 weeks without any science, without any data behind that decision.
Yeah, I agree. Yeah.
And so, as you know, Nels, a lot of rapid home test kits are going to come out to look for antibody in a pinprick of blood. I wish they were somehow reportable. For example, if you could somehow use your cell phone to report the data to a central database where people could then say, oh, look, we can get a sense in the whole country who's immune or not. And there are plenty of platforms that can do that, right?
Yeah.
And I hope some of them at least can do that, because that would be great data to have on a large scale.
Totally agree, Vincent. And I think Trevor Bedford, one of the fellows in running the next strain, has talked, I don't know if he's linked it completely to the serological testing, but certainly the cell phone, sort of the anonymized, tracing the movements to the virus test on the virus testing side. But I agree with you. If you could further link that to the serological testing, now you've got the level of information sort of that I think that meets the challenge that we're up against right now with the way this virus transmits in some, 80% very asymptomatic or pre-symptomatic cases. Even if you become symptomatic and have a bad encounter, the horse might be out of the barn in terms of the contacts you've already had 5 days, 10 days before that. And so, I agree. I think we need kind of a modern response here, in order to come through this in a way that we could reopen in an organized in a way that wouldn't get us back into, potentially even deeper trouble from a public health standpoint.
Yeah. We don't want to be congregating before the right time. Otherwise we'll just have another outbreak, right?
Yeah. Agree. Yeah, exactly. All right. Well,
we're almost done, right?
Yeah, right. We're kind of an hour plus into this. Should we move to the mailbag?
Why don't we do that and then some picks? I have office hours at 4, so.
Okay, perfect. Yeah, I think we can get through.
That'd be fine.
Relatively quickly here. So I'll open the mailbag here with just 2 letters today. So the first one is from Steve. Steve writes, "Hi, Vincent and Nels. I must admit that I can't get my head around how anything could accidentally make a protein and then learn how to build useful things with it. Maybe amino acids just started out accumulating in the membrane and gradually self-assembled in collisions with each other." So Steve is referring to this. I'll get back to the letter, but just to put in some context, he's referring to our pre-coronavirus episodes, TWiEVO, episode 51, the starting point paradox. So here, we looked at this really cool preprint from Dan Toffick's lab on deep protein evolution, right? So how did the ancient proteins sort of emerge? And so that's where Steve is, I think, raising a good point. It's really mind-boggling to think about sort of the diversity of modern proteins and how these things could start in the beginning. And Steve, I'd point you toward, I think we might have mentioned it in the episode, if you haven't already, towards some of Dan DeMaio's work, a virologist at Yale, who's pointed out how even really simple collections of amino acids, these oncoproteins or cancer or sort of transforming proteins that can cause cells to sort of dysregulate their cell growth to sort of stay open in a sense as a virus factory. Some of these viruses deploy really simple proteins with just a couple of amino acids, and then it sort of gives you the building blocks or sort of an example of how complexity can come into the course of protein evolution. But I agree. It's like completely mind-boggling as you try to rewind the tape back, 1 billion plus years to how the biodiversity of proteins sort of came to be. Steve goes on, "Anyhow, I just played your listener pick. Stirring sounds and brilliant video, but the lyrics rather get in the way and sound like they were made by one of the random new age gobbledygook making computer generators that you can find online. It reminded me of this similarly evolution-inspired Moody Blues track from a half century ago that readers and listeners might like to compare. If there is going to be a regular evolution tracks spot on TWiEVO, I like that. We might have to discuss that in some more detail. It's a shame that the complete track doesn't seem to have a suitable video. This one breaks off pretty early, but the music really provides the pictures in any case. And Steve provides us with a YouTube link that we'll put up. The weather here is now uniformly gray and damp, but the record air pressure is now back to a more respectable 1,015 millibars. First great tit songs of the year cheer things up too, with the birds emerging in spring. All the best, Steve, in Luton, England, where they've had some real weather. So yeah, that listener pick, this is from Courtney. I just went back and before our episode and took a peek at this. This is a Finnish symphonic / folk metal band that Courtney shared a couple of videos with us called Nightwish. Mm-hmm. And so we've got some some evolutionary music critiques here coming in, which is great. It's good. It's a tough genre. I'll admit to that, but I really do like this idea of evolution tracks, or sort of how does evolution emerge in different sort of musical ways.
Yeah. Or in anything besides life, right?
Yeah.
You know, art, music, fashion, whatever. It could be interesting.
I agree. So maybe we'll plumb a little bit deeper on sort of the connections between evolution in music going forward. In terms of the, I'm trying to remember, it's not quite, my recollection is a little bit vague on how we got on to the sort of, I think we were talking about Swedish heavy metal somehow, and I kind of made a clumsy statement about that. And then Courtney put up this really nice set of links to Nightwish. Just reminds me, I have another colleague who, he was a grad student in Mike Emmerman's lab when I was a postdoc at the Fred Hutch. He's now, his name's Simi Tareen. And in addition to he's now working at a company, but in addition to being a virologist, he is a massive heavy metal, music fan. And in fact, he even scores musical compositions for horror movies. And so I'd put up, maybe I'll grab Simi at some point to maybe help curate our,
That's cool.
Yeah. Our evolution sort of themed, evolution tracks with a kind of heavy metal, bent to it. So anyway, thank you, Steve, and glad that, protein evolution episode, ancient protein evolution episode sort of piqued your interest, and you're in good company when it's to put our minds around 1 billion plus years of evolution is not a trivial matter.
For sure. All right, we have one from Nathalie who writes, Dear Nels and Vincent, firstly, hope to find you both healthy and sane in these troubled times. Secondly, thanks for doing such a great job broadcasting scientific papers to a big audience. I love how you explain and subtract important evolutionary concepts in each paper. I'm a postdoctoral researcher in Groningen, and because we are not allowed to go to the lab anymore, and there is, of course, good reason for that, we have increased the frequency of our journal club. I'm writing to you because my colleague picked a very interesting article for it. Thought you might find it interesting. It's from the Murray Lab from Harvard and explores how cells might adapt to perturbations in things essential to them, such as, in this case, replication. And then she provides a link to that paper. Stay healthy, Nathalie. So thanks, Nathalie. Thanks for the compliment in the link. This is an eLife paper. The evolutionary plasticity of chromosome metabolism allows adaptation to constitutive DNA replication stress. This could be interesting, Nels.
Yeah, it's really interesting. So this is from Andrew Murray's labs, and he's well known as one of sort of the pioneers in some of the cell biology behind chromosome replication. So the notion of error catastrophe in microtubule, the building of microtubule polymers and the breaking of them, and then related work. What's really exciting is Andrew's lab has moved pretty decisively into the field of evolutionary cell biology. And this is a great example of it. I really admire this paper. There's another one that actually has been on my radar. Obviously, we've gotten a little sidetracked here, and it might be sidetracked for a while. But when we do sort of return to the emerging great papers in kind of modern evolutionary biology, I think adding this one from Andrew, and then he's a co-author on another paper from his colleague Ethan Gardner, also using yeast, but to look at how actin proteins have evolved as part of the activity, and some of the surprising sort of both the flexibility of some of these really well-conserved proteins, but also how you can spin out different kinds of functions by taking an evolutionary view. And then in particular, I think what really kind of puts this kind of work really sort of distinguishes it in some ways is that they're taking a very much a cell biology or experimental approaches, and taking advantage in this case of Saccharomyces, the baker's yeast, the long-standing genetic model system, in order to do real-time experimental evolution. And so we'll get to those papers eventually. In the meantime, actually, I'd point our listeners backwards to TWiEVO episode 22. mysteriously titled E pluribus cerevisiae. This was a really fun interview Vincent and I had with Maitreya Dunham from Genome Sciences at the University of Washington. She was telling us about all these cool kind of automatic chemostat systems to continuously grow yeast under different selective pressures and then see how they evolve in real time. And so you can kind of hear it directly from Maitreya. So Ethan and Andrew and others have been using some of those techniques. But in this case, the selective pressure is sort of manipulating or disrupting some of these core replication functions involved in chromosome segregation or in actin dynamics, and then let the yeast sort of evolve in real time and provide some answers about some of these evolutionary pathways. So really cool stuff. Cool. So thanks for bringing it up, Natalie. Okay, so then to close out on science picks, I had one. And we've talked about this in the past a little bit, but this interactive science museum that I have a personal connection with, the Science Mill, this is in the hill country of Texas. My dad, who's now a retired neuroscientist and stepmom, who had some med tech businesses that she sold and retired. They've been running for a little over 5 years now, this in Johnson City, Texas, the Science Mill, Interactive Science Museum. Of course, they've had to shut down operations given the coronavirus situation, even in rural communities. And so they're scrambling to try to keep sort of a skeleton crew operation going and to provide some resources that might be of interest, I think, to our listeners and beyond. So, of course, we now have a lot of friends, family, colleagues, other folks who are parents with young kids and spending a lot of time at home with each other. And so the Science Mill is beginning to offer this program, and I'll have the link up here for my science pick, which is they're calling the Explorer Zone. And they're describing it as an online learning hub featuring videos, hands-on science and interactive games. Each week, the Science Mill staff will take you on a virtual field trip on one of the museum's 50-plus exhibits. And then they also include activities that you can do together to explore some of these connections to STEM careers, science, technology, engineering, math.
Cool.
Check it out. If you are, a parent with young kids or a kid at heart, I think some really great resources that they're putting together. And to me, this sort of, it's a nice example of all of us trying to do what we can under pretty tough circumstances and contribute where we can. And so, I'm sort of, very peripherally involved in it, but wanted to, support and lift that up.
Now, they have a cool webcam. I'm watching a couple of tortoises eating lettuce live.
That's great. Yep. Yeah, you can see in the first episode they did too is an aquaponics exhibits where you can see the koi fish and the plants growing together cool. And describing the science behind it. Chewing away.
They're just chewing away.
Yeah, the African tortoises have a really great setup at the Science Mill.
Obviously, someone had to put some food there for them.
That's right. So,
but they're streaming through Twitch, and I say I have done 2 live streams at Twitch, which is largely a gaming site. But they invited me to do 2 COVID-19 live streams, and we got tens of thousands of people listening in. So it was very cool.
Wow, that's great. Yeah, we've been brainstorming a little bit at the Science Mill about how do we, and you and I have talked about this a little bit before too, is how do we bring viruses into natural history museums or interactive museums? And so we're in just some preliminary discussions about how we might revisit that as this plays out in the situation. And then as things reopen, The Viruses of Our Lives is sort of an early working title for thinking about how we might share some of not just the kind of pandemic and negative sides of this from a public health aspect, but also just the richness of biology and the kind of all of the connections of how viruses are really important in our lives in many different ways, both from a health standpoint, but also in many positive ways as well.
Yeah, I have some ideas if you want to consult sometime.
I would love to. And I should say Rich Condit and his wife have been to the Science Mill since they're somewhat in the neighborhood. They're in Austin, Texas, about a hour or so drive away. And so it'd be really fun to think about teaming up in some way, shape, or form going into the future. How about you, Vincent? What's your science pick of the week?
This is an interesting site from the Lipkin Institute. It's called COVID-19 Treatment and Vaccine Tracker. So they're tracking developments of vaccines and antivirals, antibodies, cell-based therapies, RNA-based. There's a nice pie chart on the front, and then you click on the orange bar and you can go through each one individually. You know, what it is, who's making it, where it is currently, where it's getting funded, what's the next step, the timing, and they have links to the sources. I think it's a really nice compendium of everything. And there's a ton going on. Let's see.
Wow. Yeah.
We go all the way to the bottom. Well, there are 84 different therapies, and vaccines, there are 50 vaccines in development.
Yeah, it's really amazing.
It's amazing.
Yep. Great resource.
It's cool.
Look forward to it's incredible too, you know, just the kind of on the ground response. So we just locally at Utah had a call for seed grants related to SARS-CoV-2. And, you know, I think the crew that put it together thought, oh yeah, we'll get a dozen applications or so. There were 80 people or 80 groups that applied for a seed grant.
Yeah,
and this is coming from different departments all over the university. It's not just virology. It's also all of the associated, you know, this kind of touches every part of our lives. And so, yeah, no, wow. That's really, I'll take a closer look. Thanks for pointing out all of this energy and action that's going in this direction.
All right. That'll do it for TWiEVO 53. You can find it on any good podcast player. Show notes are at microbe.tv/TWiEVO. Please consider subscribing on your podcast player so we know how many people are listening, and you get every episode every month as we release them. Of course, they're all free. If you really like what we do, consider supporting us. You can go over to microbe.tv/contribute, and there are a couple of ways where you can donate as little as $1 a month, help with our expenses. And of course, questions and comments, TWiEVO at microbe.tv. Nels is at cellvolution.org. Over on Twitter, he's L Earlybird. Good to talk with you again, Nels.
Yeah, great to talk to you, Vincent, and strength to your voice as you continue podcasting far and wide and showing up on CNN. And thank you for all you're doing. Really appreciate it.
You're welcome. I'm Vincent Racaniello. You can find me at virology.blog. Music on TWiEVO is by Trampled by Turtles. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Till then, stay curious.